Okay, well, it is now officially afternoon, so I will get started. Today we have another four outstanding trainee speakers. Hopefully you guys were able to join us last week. Nobody disappointed, and I'm sure that's going to be the same way today. I'm going to briefly introduce each of them and then turn off my camera. And then after about 9 minutes, I'll turn my camera back on. And after about 10 minutes, cut you off for a little bit of Q&A so we can have some discussion about the excellent work you're all doing. So first off, we will hear from Hannah Cutshall. Hannah received her BS in molecular and cellular biology at Vanderbilt before entering medical school here at UAB, which she's set to complete, it looks like in June. So good luck to her. She has several interests, including one that she won't tell us about today, the image or molecular and genetic epidemiology study of multiple myeloma. She also has several presentations at the national level, including last year at the American Society of Hematology. On a side note, I noticed on her CV that she's actually a runner after my own heart, but unlike me, she's actually won a marathon, having won the 2015 Nashville Marathon. So congratulations to her for that. But today she's going to tell us about another research interest she has entitled Impacts of the Virtual Landscape During the COVID-19 Pandemic on the 2020 Application Cycle. Hannah, the floor is yours.
Thank you so much for that introduction. Let me just get my slides shared here. Okay, into presenter mode. All right, can everyone see my slides and hear it okay? All righty. Okay, so this presentation I'll be giving today is on the impacts of the virtual landscape during the COVID-19 pandemic on the 2020 application cycle. So a little background on this project. As of May 11th, 2020, the AAMC released the recommendations that all residency interviews be conducted virtually and that away electives be discouraged. So this project is actually a two-part project that we've been working on. The first part was on the pre-interview virtual interactions through social media usage and virtual open houses. And the second part, which I'll be presenting on today, is on the impacts these had with the virtual landscape on the 2020 application cycle. Okay, so for our methods, this is for that part one study, which was actually presented at last year's grand round. So if you've heard a little bit of it, sorry about that. But the first part methods was the 138 total pathology residency programs were analyzed. All programs were analyzed for use of Twitter, Instagram, Facebook, or other virtual opportunities through the departmental and residency accounts. And all data on these account types were obtained as of October 30th, 2020. Okay, so the methods part two, which is what I'll be presenting on today. This part of the study was taking that data that we got from our previous study and then conducting an anonymous survey sent out to all the programs who had engaged in social media and pre-interview virtual interactions that we had seen in that 2020 application cycle based on our previous study. So the survey was distributed to the public social media accounts that were used by these programs, as well as to the program contact email listed on their department website, and that the survey was also sent out to matched applicants who used the social media tag #PathMatch21. And then all of the results were compiled through the Qualtrics system, which was the system that was used to construct this survey. So as you can see in table one, here is our distribution method. So the main thing was through email, and that was, again, just what was listed on their departmental websites, typically either the program director or program coordinator. We also used social media engagement, so Twitter, Instagram, and Facebook, and that was all to the public accounts. So primarily Instagram on that one, as most of the Twitter and Facebooks were private. And then you can see here our matched applicants outreach on Twitter. Okay, so our results, let me see if I can move this bar out of the way. Results that we saw were who responded and from where. So our responder classification, it just shows in this table who all responded. So program directors, we had 6, associate program directors, we had 5, faculty 6, residents 17, and then those applicants 19. And then this figure here shows the region that these responders were from their home institution. So as you can see, most are from the southern region, followed by northeastern in the second. We had a few from the central area and 1 from western, and this was just based on what state they responded, their home region was. And that is broken up into the US Census of what region they fall in. And that kind of correlates with what we saw in the first part of our study, being that more social media outreach was used in the southern and northeastern regions as well. Sorry about this bar. So results that we saw, just impacts from the program perspective. Our big finding was that yes, the programs did feel that the applicants missed out on experiencing the program, with 80.7% responding that yes, they did feel that the applicants missed out on experiencing the program. One other thing we saw was we asked about number of candidates invited to interview and interview days offered. And 56% responded yes to both of those, that they invited more applicants to interview and offered more interview days. And then also we asked, kind of interesting from the applicant side now, we asked if it affected the rank position of any applicants, and we saw that 16.7% did respond that yes, they moved applicants up on their list based on these pre-interview virtual interactions. However, the majority at 79.1% said it did not impact their rank positioning. We also asked if they would plan to continue to utilize these pre-interview virtual opportunities if we went back to a normal in-person interview cycle. Most, 70.8%, said unsure, and then 29.2% said yes, they're a hybrid model of both in-person and these virtual ones. But nobody said no, that they would not be interested. Okay. So results from this one's from the applicant's perspective. So this one, our main thing is that we saw it did actually impact their ranking of programs. So we saw that 56.6% said they ranked programs higher based on their pre-interview virtual interactions, whether that be open houses, social media engagement, or such. Saw 17.4% ranked the programs lower, and 13% ranked programs that they otherwise would not have ranked. So that was very interesting. We asked them the interview preference of if they would prefer in-person, virtual, or unsure. We saw a pretty even split on this one, was 29.4% for in-person and virtual both, and then 41.2% for unsure, which, given this is from the applicant perspective, so they would not have had a previous in-person exposure to interviewing to compare it to. Okay, so then one of our other findings here is more specifically on the interviewing. So we see figure 2 and figure 3 here. Figure 2 shows our number of programs the applicants applied to, while figure 3 shows the number of interviews that they actually completed. So as you can see, 42% of our surveyed applicants applied to more than 50 programs, with 3 applying to greater than 90, which is a little crazy to me. But figure 3 here shows that out of all these applicants surveyed, none completed greater than 30 interviews. So this kind of could hint at maybe a little bit of over applying on the applicant side, which I guess programs did mention that they were offering more interview days and inviting more applicants to interview. But so this could be a possibility of over applying due to not getting to do away rotations and seeing the program that they may have been interested in, or feeling that they didn't have the proper letters of recommendation from away rotations possibly. Or just an ease of going to the different programs with not having to actual travel to these programs, they can apply to more. But just saw that a possibility of over applying with these results. So what about UAB? Well, I have 2 years here. So all of our previous results were only from the 2020 application cycle, but now that we're in a second year of virtual interactions, I thought I would include this year, and also a little biased being an applicant this year as well. So the 2020 side, we had 4 virtual open houses and also had faculty testimonials on Instagram. 2021, a few things changed in that now it's UAB Hearsink School of Medicine. They also this year had 3 pre-interview virtual open houses, 1 open house for the interviewees, an updated tour video and testimonial video, and drone footage of the faculty group. And I will say, as an applicant, they had a very interesting open house. They made it very fun, had games, did scavenger hunts. It was a great interaction, even not getting to actually be in person. And one thing that was very unique about UAB is they also in 2021 have developed a softball team called Softball Team NOS. And one of the pre-interview virtual interactions they did was actually a live Q&A session from the softball game. So they got to show everyone the softball game. They got to see how the residents interacted and worked as a team, and also got to have their questions answered as well. So that was a great experience and just something a little bit different, but that was that. Okay, and here are my references. And that is all. Thank you so much for the opportunity to present at Grand Rounds, and I can answer any questions.
Perfect, thank you, and thank you for staying right on time. So it looks like you have one comment from Brandy. Both yours also included an institutional sponsored diversity open house. So I really like that we were able to hear how this has progressed. I know COVID has hit various things in many different ways, and it's really impressive to me to see how you guys have been able to pivot and adapt to this. It's really brought in new avenues, which I think you've really maximized, and that's nice. Dr. Harada asks the question, any speculations on other specialties?
Yes, so this project was actually part of a, we called it a consortium of other specialties as well. So they are working on getting the final paper published with all of the results, but that included 24 other specialties. And this was one of the projects within that consortium. So we are still waiting on the final publication on that one to have all the data fully released, but that is in the works.
And I'll echo Christina's point. It's really good to hear that this is paying off. One of the things going virtual, including my role is organizing this Grand Rounds, I often worry what impact does the virtual have, and it's good to hear that it's coming off positive in your end. So do we have any other questions or comments for Hannah? Okay, so hearing none, thank you very much for that.
Thank you, and I'll stop sharing.
Perfect, thank you. So next up, we will hear from Dr. Prachi Bajpai. She initially received both her master's degree first at the CSJM Kanpur University in India, and then a PhD at the Indian Institute of Toxicology before coming to UAB, and she is currently a researcher in the laboratory of Dr. Upender Mani. She has an impressive 19 manuscripts, including contributions to one published just last month in Frontiers of Medicine, and has another 6 listed in review or preparation, so very productive. She has well over 20 national and international meeting contributions. And what I found interesting about her CV is that she's also contributed to several projects that have actually garnered lay press attention, including some from the Daily Mail on reversing aging. So if you're feeling a little old, I think we should talk to her about how to feel a little bit younger again. But today she's going to tell us about reducing regorafenib toxicity, tumor progression, and metastasis in colorectal cancer by combining with JAK and HDAC inhibitors. So Dr. Bajpai, the floor is yours.
Thank you, Dr. Wendy. I'd like to start my presentation by thanking the Pathology Trainee Committee and Dr. Mani for presenting me an opportunity to display the work that I have been doing for the last 1.5 years in this lab. Basically, we will be targeting the toxicity for the regorafenib by a dual inhibitor, which is JAK-HDAC. And the take-home message would be how the colorectal cancer tumor growth and metastasis could be curbed. Sorry, it's okay. The study focuses on a regorafenib, which we all know is a multiple tyrosine kinase inhibitor, and quite evident because of its toxicity. And the work that I'm doing is basically a preclinical testing of the combination of this novel drug combination of JAK and HDAC with regorafenib with in vitro models and in vivo with CRC PDX and HT29 experimental metastasis model. I'll also touch base with the mechanism of action. So we are all aware of the colorectal cancer progression that when pathologic lesions appear in the normal epithelium, it advances to adenoma and then carcinoma. And then It metastasizes to various distinct organs like bone, brain, liver, where primarily or most of the metastasis lesions are seen in the liver, shown here in the schematic. So in the early stages of colorectal cancer is the tumor could be resected, but the late stages, stage 3 and 4, they are treated following the NCCN guidelines, which are presented here, as first line, second line, and third line therapy, where regorafenib is a treatment of choice when the patient recur or relapse after the first line of therapy. Having said that, after a decade ago of US FDA approving this drug for metastatic colorectal CRC, which were refractory to the first line therapy or intolerant to the standard chemotherapy, the caveat that remains or the major concern is the adverse effect. Here I'm presenting a schematic, or in fact a summarized table for phase 3 clinical trial for 4 major clinical trials. We see there is a survival benefit, a marginal survival benefit, I would suggest. That's what it's reported. But 25% of the patients are withdrawn or discontinued due to the regorafenib toxicity because it's of hand and foot skin reaction, hypertension, diarrhea, and fatigue. This is kind of a dual-edged sword, and that's where our prime focus is, how the adversity of regorafenib could be decreased. This led us to a novel JAK-HDAC inhibitor. We all know that individual inhibitors for JAK and HDAC are approved by FDA for various treatments, but the novelty for this study is the combination of the JAK and HDAC. We all know that JAK-STAT pathway is haywire in the colorectal adenomas, and therefore, one additional thing that I wanted to point out is that concurrent inhibition of JAK sensitizes tumors to the HDAC inhibitor, and possibly that led to the rationale of development of this hybrid molecule. A very interesting work recently published in 2019 in a preclinical settings where they have shown the efficacy of this hybrid drug in the CML and other hematological malignancies. So the rationale for our work was to see its efficacy in a solid tumor, and because the lab focuses on colorectal cancer, that was the take-home efficacy model. So now we are moving towards the result with that brief introduction. The prime focus, as I just mentioned, is to address the toxicity for regorafenib. Now, how that was addressed is we took normal colon cells and took various concentration-dependent doses of individual drugs and the combination, and selected a drug concentration which was not inhibited in the normal colon cells. So this somewhere around 500 is what we see is a subtoxic dose, which doesn't cause any effect to kill the normal colon cells. And this 500 nanomolar, when I took in 6 different cell lines the treatment regimen, we see that there was marked inhibition in the colony formation, be that single and much pronounced in combo. So I'm sure this slide conveys the point that the 500 nanomolar concentration, or that subtoxic the dosage that I'm taking, does not have effect on the normal colon cell, but it's quite pronounced to kill the colorectal cancer cells. Now, because we know the regorafenib is a multi-tyrosine kinase inhibitor, before we go ahead with the kinome profiling, which shows how the different kinases are modulated, the Western blot here shows the efficacy of individual drugs. So here, what I'm trying to convey is JAK-HDAC inhibits phosphorylation of STAT3, which we see here with individual combination of JAK and the combo. So definitely we know that the phosphorylation of the STAT is inhibited by this drug, and the regorafenib take-home message by inhibition of phospho-ERK as evidenced here. So once we know that everything is up and running, the inhibitions of individual drug targets that we showed by phospho and phosphostat, we went ahead for the kinome profiling. And here I'm showing the network hub for different kinases that were altered. Anything which has a blue dot shows that the kinases are inhibited. So clearly, I'm sure the audience would appreciate that most of the kinases were inhibited in the combinational treatment, unlike the individual treatment of HDAC and regorafenib. And in addition to these multiple kinases that were inhibited, we found that several cyclin-dependent kinases were the main prime targets. So we further evaluated our effect on cell viability. This is the data for MTT, clearly indicating the fact that the combination treatment in 3 different cell lines is more pronounced in combination, like it went down to 35% if we compare the differences here versus individual differences here. So marked reduction was observed in the combination. As I clearly mentioned, the cyclin-dependent kinases were altered. This shows that there was G0, G1 arrest in the combination in a delayed S phase, which shows the proliferation was reduced, and different markers for apoptosis was also decreased, as we see here. So we led to the complete holistic picture to understand what's exactly happening. We did RNA-seq, and this is a snapshot for the different pathways that have been altered. And we see increased PUMA the apoptosis of several pathways were induced, where PUMA is one of the major targets that we see here. So this further we validated in 4 different cell lines models. We see concomitant increase in PUMA in all the different combinations. Moving forward to the efficacy of the drug combination in vivo settings, we would like to share our data with CRC-PDX model. The combinational treatment shows marked decreased expression in tumor weight and size as evidenced here. And here, this is the H&E staining for all those tumors with single and combinational treatment. And I'm sure I'm able to convey the message with this highlighted thing that which we have more necrotic area in the combination. The Ki-67 staining further confirms that the proliferative index went down with the combination. Now, the study is incomplete without addressing the toxicity. Now, this is a serum analysis that we have done with the CRC-PDX models, and we see most of them were within the reference range. These parameters are basically a take-home message to understand the liver and the kidney functions. Another step forward is to show the metastasis, how that was curved. So this is a bioluminescence imaging with HT29 cells, and it clearly indicates that in combinational treatment, the metastasis regressed. H&E staining further corroborate with the same findings that we saw in the first slide, that combination treatment have much smaller tumor, unlike the vehicle and single treatments. This is for this H&E is for regressive metastasis in kidney, as highlighted here. And the further serum analysis again with this model showing that all these parameters for liver and kidney were within the reference range, clearly indicating there was no toxicity, and still it was able to curb the metastasis in the HT29 model. So in snapshot, the conclusions that I would like to convey to the audience is the combination of this regorafenib and HDAC inhibitor. It reduces cell proliferation and colony formation and increases cell cycle arrest and apoptosis in CRC cells. It also reduces the tumor growth in CRC-PDXs. Tumor xenograft model also conclusively showed that it regresses metastasis. Kinome profiling proves a point that multiple kinase activity are inhibited in the combination. And this is through inhibition of STAT3 and ERK and induction of PUMA. And whole transcriptomic profiling shows apoptosis signaling is modulated. The study would not have been complete if I would not have got support from very cooperative lab members and primarily to a mentor, Dr. Pinder Mane, for his guidance and supporting this work generously and believing in the data and more so in me. Dr. Kim for generating the PDX's model, members of Mane Lab, Dr. Sumit Amar, and Dr. Varambali and Darshan primarily for bio RNA-seq analysis, and very supportive Dr. Miller's lab for RNA-seq. I appreciate Dr. DeFala's expert comments and image evaluations and helping me analyze the data. Dr. Kushman and Dr. Paluri for critical discussions, and Dr. Mane's collaboration in Morehouse where the serum analysis was performed. Thank you.
Great, thank you so much. Since we're running a little bit ahead of time, we still have time for one or two questions. Will people either type it in the chat or turn on their cameras? I'll ask the first one. In your Kinomic analysis, I noticed AKT, and I know AKT is very involved in cancer as well, but in my mind, it's always insulin signaling, right? So is there any concern? I mean, you've shown very clearly that the tumor's growing less, which is great. Is there any concern about pushing the patients toward a diabetic state or insulin resistance?
So let me show you the figure where it's not just AKT is also one of the factors inhibiting by JAK-STATs. It's not it's something which we anticipate, but not a non-specific effect that we are seeing. And definitely what you are saying, we know that JSK beta is inhibited with the regorafenib. So with my understanding of the serum profiling and other things, my anticipation is it did not go haywire. So I don't think there should be an issue, but yeah, that should be a good point to look into the sugar level as you're mentioning. Yeah, but AKT is definitely a target for regorafenib. So its inhibition should not be an issue.
Okay, thank you. Are there any other questions? Okay, well, thank you. It was a very clear presentation, so I think you answered them all during it. Thank you very much. Okay, so our third speaker today is Dr. Elizabeth Evans. She initially trained at Wesleyan before receiving her medical degree from the University of Virginia. She then went on to complete her residency at the University of Oklahoma before we were lucky enough to get her at UAB for her fellowship, which she started back in 2021. She has several publications, including one recently accepted in Current Oncology Reports. And in addition to that, she has contributed presentations to several meetings and other grand rounds. As well as doing all that research and clinical training, she has also initiated efforts for education and quality improvement, which I think is also very important for the school overall. However, today she's going to tell us about activins in ovarian cancer. Dr. Evans, the floor is yours. And you are still muted. Okay.
Sorry about that. Thank you so much for that introduction. And thank you to the Department of Pathology for allowing us this opportunity to discuss the work I've been doing for the past couple of months. Before I get started, I want to just acknowledge the mentorship I've received from Dr. Maitri Karthikeyan this year, as well as from Dr. Rebecca Arend, who really helped guide me throughout this entire process. All right, so for some context, in the United States, ovarian cancer is the second most common gynecologic cancer, with more than 20,000 cases diagnosed annually, portending about a 1.3% lifetime risk to women per year, which correlates to about 1 to 72. Ovarian cancer is the most common cause of death by gynecologic malignancy and the 5th most common cancer-related death in women. Ovarian cancers are classified typically by their cells of origin, with the most common subtype, epithelial ovarian cancer, arising from the epithelial lining of the ovary. These can then be further stratified by their histology. Effective screening and early diagnostic protocols remain elusive, and so the majority of epithelial cancers are unfortunately diagnosed at advanced stage. At this time, the standard of care remains cytoreductive surgery, followed by platinum-based systemic chemotherapy. Unfortunately, many of these tumors do occur with a high rate of resistance and poor response rates to the available therapies at that time. A better understanding of the biology that underlines ovarian cancer development and disease progression can help identify therapeutic targets for future use. So, what exactly are activins? Activins and inhibins are a subgroup of unique ligands within the TGF-beta superfamily. Both arise from dimerization of a shared pool of subunits. Genes encoding in INHA, INHBA, and INHBB produce monomeric alpha, beta A, and beta B subunits respectively, while inhibins are heterodimers formed by linkage of the alpha subunit to a beta A or a beta B. Activins, which will be the focus of today's talk, are formed by homodimerization of the beta subunits. Well, first identified within gonadal tissue, with the root of their names arising from the polarizing influence of activin and inhibin of the hypothalamic pituitary gonadal axis, the presence of these proteins has now been described in a wide range of human tissue types. Activin plays a role in many physiologic and pathologic processes, with implications both on tumorigenesis and metastasis. Activin signaling is accomplished via two-step interaction with the serine threonine receptor complex and intracellular SMAD signaling proteins. Activin directly binds to the type 2 receptors, active receptor type 2A or B, which then recruit and phosphorylate the activin type 1 receptors ALK4 and ALK7. This in turn results in the phosphorylation of secondary messenger SMAD, most commonly SMAD2 and 3, which can complex with SMAD4, and then translocate the nucleus to affect gene transcription. Evidence has been accumulating for a role of activin as a biomarker of malignancy, with both potential diagnostic and prognostic implications. Several exploratory analyses have been done looking into INHBA gene expression profiles in different solid malignancies. INHBA, which as a refresher encodes the most common activin subunit, the beta A subunit, is frequently used as a representation of activin as the ligand as a whole. Level of INHBA expression has been found to be significantly correlated with disease-specific survival outcomes in many cancers, with prior work in Dr. Kirthikian's lab presented here on the left, demonstrating a high correlation between elevated INHBA and INHBB expression and worse prognosis in many solid tumor types. In particular, they found increased INHBA expression in ovarian tumors, which was correlated with worse mortality with a hazard ratio of 1.16. A similar pattern was noted with INHBB, but this did not reach statistical significance. And so because of this, my work is largely focused on the beta A subunit over the beta B. In all, the role of activins in tumorigenesis appears to be both site and context specific. And so my work this year is focused on characterizing this further, specifically in an ovarian cancer model. We first have started with RNA-seq. So using RNA sequencing data previously obtained by Dr. Rebecca Aaron's lab, we performed analysis of ovarian cancer tissue, comparing them to a benign cohort or non-cancerous cohort. In all, we had ovarian samples from 62 patients, 52 of whom had malignant disease, all high-grade serous ovarian cancer, the most common form of epithelial ovarian cancer, and 10 with benign pathology. In addition, cells derived from ascites were analyzed from 24 more patients. We began by assessing gene expression in solid tumors, comparing them to benign and to ascites, and then further stratified the solid tumors by platinum chemotherapy sensitive and platinum chemotherapy resistant. What we found, consistent with the prior literature, was that activin gene expression was elevated within malignant ovarian tissue when compared to the healthy cohort. Both INHBA and INHBB, the genes encoding for the two subunits of activin, were significantly elevated in malignant tissue when compared to benign. This pattern persisted when stratified by platinum-sensitive and platinum-resistant tumors. Regarding the genes encoding the type 1 receptors, ACVR1B, which encodes type 1 receptor ALK4, was found to have a nearly twofold greater expression in tumor versus benign tissue, while ACVR1C, which encodes type 1 receptor ALK7, trended towards a lower expression in the tumor compared to benign tissue, but this did not reach statistical significance. No significant difference was noted between the platinum-sensitive or platinum-resistant cohorts here either. So several members of the TGF-beta superfamily, including Activin, have been found to participate in invasion metastasis of many malignant tumors, with most of the data actually looking at the influence within colorectal and prostate. We decided to look at the solid tumor versus the ascites cells and found that they significantly differed in their activin signaling expression profiles. Interestingly, when comparing solid tumor to the ascites, which can be considered representative of a metastatic subpopulation of this cancer, genes encoding for both activin beta A subunit and its type 1 and type 2 receptors were upregulated, meaning that higher relative levels of gene expression were seen in the primary lesion when compared to the ascites population. More specifically, genes encoding for the activin beta A subunit, as well as the type 2 receptors, were upregulated in solid tumors when compared to ascites cells. Altogether, this suggests a potential difference in expression pattern at primary and metastatic sites, which has not been well characterized in ovarian cancer and which we wish to explore further. Of course, RNA sequencing of bulk tumor burden is non-specific in the sense that you're evaluating both ovarian cancer cells as well as other components of the tumor microenvironment, including immune cell infiltration. Members of the TGF-beta superfamily have been noted to play a critical role in the maturation and activation of both the innate and adaptive immune systems. Activin, in particular, has been implicated in the conversion of CD4, cytotoxic CD4 cells into the more immunosuppressive FOXP3 positive T regulatory cells, which are defined by their immunomodulatory role. In several cancer models, Tregs play a central role in assisting tumors evade immune destruction. Activin, through its influence on the immune phenotype, may thus help promote a more pro-tumor microenvironment, either locally at the primary tumor site or distantly at the site of metastasis. The two aims of our project broadly are as follows. Aim 1 was first to simply better characterize the expression profile within ovarian cancers. We wish to perform immunohistochemical analysis of the inhibin beta A expression in primary and metastatic sites of several epithelial ovarian cancer subtypes. Expression will then be scored with assistance of our pathology colleagues and correlate with the expression of cytotoxic and immunosuppressive tumor-infiltrating leukocytes. Our second aim is to better characterize the baseline activin signaling within ovarian cancer. To accomplish this, we'll be utilizing a panel of both human and mouse cell lines. Cells will be treated with both recombinant activin as well as an activin receptor ligand trap to assess the influence on cell growth and colony formation in both chemosensitive and chemoresistant cell lines. To mirror the metastatic potential, survival will be assessed in both 2D and 3D culture. Lastly, we will assess serum and ascites activin levels in samples obtained from ovarian cancer patients as well as a healthy patient cohort. Our TMAs were created in collaboration with the tissue biorepository here. On the screen is a representative sample of one of our TMAs, this one composed of mucinous tissue. In all, we obtained tissue specimens from 43 patients with epithelial ovarian cancer, 22 of whom had mucinous, 21 had clear, and 40 had high-grade serous. In 38 of the 40 patients, we were able to obtain matched metastatic tissue as well. And we will extract further demographic and clinical data from the EMR to allow for correlation with prognostic outcomes. Antibody optimization is underway, with further optimization achieved in conjunction with the Pathology Core Lab. Tissue scoring will be performed in collaboration with our pathology colleagues. So to assess activin responsiveness, we've been utilizing a cell culture model, which encompasses a panel of somatic and germline mutations commonly seen in epithelial ovarian cancers. More specifically, we'll be using mouse and human cell lines, which harbor mutations in tumor suppressor genes P53 and BRCA2, as well as several human lines mimicking chemosensitive and resistant states. To assess signaling response to activin, cultured cells will be treated with recombinant activin. A more response rate was seen in the human cell lines of CAR3 and HAE, with the greatest relative effect found at 1 nanogram per milliliter, with signaling responses plateauing at higher doses. In the mouse model, D8, P53, and BRCA2, higher baseline levels were appreciated in both lines, with less responsiveness to the addition of recombinant activin. These experiments will be repeated with a commercially available activin ligand trap, as I mentioned, to assess the effect of activin suppression. We then utilized PCR to assess activin receptor expression in our selected cell lines to see if this could account for the difference. Both human and mouse lines were notable for a relative absence of activin type 2B receptors, but these models differed in their expression profiles of the type 1 receptors ALK4 and ALK7. Specifically, the human-derived OVCAR3 and HAY lines expressed ALK4 predominantly, while the mouse ID8 lines did not, expressing ALK7 instead. This is consistent with our prior RNA sequencing data, which, while not reaching clinical significance at that time, demonstrated a relatively lower expression of genes encoding for the activin type 2B receptor, as well as the ALK4 receptor. Given that the difference in ACVR2A gene expression was significant in the solid tumors versus the ascites, we will then assess for differences in receptor expression in the 2D and 3D culture. In upcoming months, we wish to complete our IHC analysis of the immune infiltrating cells within the ovarian cancers and examine their correlation with activin. In addition, we'll be assessing activin responsiveness in chemosensitive and chemoresistant cell lines. And given the differences in expression profiles between the solid tumors and ascites cells, we will assess the impact of activin on survival on these two populations as well. Lastly, we wish to suppress activin levels with the use of the commercially available activin ligand trap and evaluate the effects on tumor burden and metastasis. All right, thank you. I'm sorry I ran a little over, but I'm happy to answer any questions.
That's okay. You each have 15 minutes and you're still well under that, so we definitely still have time for a question or two. So very exciting work, definitely setting up a good precedent for doing the trials with the activin trap to actually see if this will have therapeutic potential. I think it's very convincing that this is the right direction to go. So definitely set the stage very well. Do we have any, I guess that was more of a comment than a question though, but does anybody have a specific question for Dr. Evans? So when do you think you'll actually get to start those initial proof of principle studies with the trap that you just mentioned, the last point on the previous slide?
As soon as it arrives.
Okay, great. So
yeah,
definitely look forward to hearing that in the future. That sounds very exciting. Well, great. Okay, well, hearing no other questions, thank you very much for that. And we will go on to our last speaker. Okay, so our final speaker today is Christopher Ramirez Livelo. He initially completed his BS at UCSD and then a master's degree at San Diego State. He then spent some time working at Illumina, which is very interesting and a very important company for a lot of the work I do. So thank you for that. He then started his doctoral career and then made a shift to come to UAB, where he's currently in the GBS program and a student in the laboratory of Garesh Melkani. He has several publications, including the very exciting information of a first author manuscript under revision at Nature Communications. Today, he will tell us about time-restricted feeding attenuates obesity-related muscle dysfunction through AMPK and purine pathways. So the floor is yours, Christopher.
All right. Thank you very much for that very generous introduction, Dr. Wendy. And with my excitement, I would love to present my topic here. Let's see here. Let me first get into the slideshow. Okay. So as was introduced, my name is Christopher Livelo. I am one of the few students in Dr. Garesh Melkani's lab under the pathology department, and it's really a privilege to be a part of this pathology grand rounds and discussing my research. And it's a very exciting topic that I'm really passionate about. The title of my talk is going to be about time-restricted feeding and how this intervention can attenuate obesity-related dysfunctions through AMPK and purine pathways. So at the very beginning, I'd like to start off with the impact of metabolic challenges, and the ones that I'm currently interested in are obesity, circadian disruption, and aging. And As you can see from these statistics, this has quite a far-reaching impact, and so many people are currently afflicted with a lot of these metabolic challenges. And furthermore, how this actually connects and plays a role with skeletal muscle function is really what I'm interested in pursuing. So skeletal muscle is very important. There's a lot of different roles that the skeletal muscle is part of. A lot of this can include glucose uptake, energy metabolism, and a lot of this can actually relate in dysfunction when combined with the proposed metabolic challenges. So what I'd like to connect and inspire upon everybody is that I'm currently exploring how circadian rhythmic timing and other potential avenues such as feeding fasting rhythms, how this can actually play a role in regaining this metabolic control under these different challenges. So there are two different external cues here. One is light, one is food. And currently what I'm working on is a feeding fasting intervention. And this basically deals with the introduction of how you time different meal times. And by doing so, you're actually able to affect the local oscillators, whether it's through the core clock in the brain, or you can actually affect the peripheral tissues. A lot of these can then move on to actually modulate gene expression pathways through these oscillations. And this can have varying effects on things such as metabolic pathways. And this is very interesting to me. And one of the schemes, the way that we're introducing this is we have what's called a time-restricted feeding condition, and we have an ad libitum feeding or ALF condition. Now, in our lab, we work specifically with fruit flies, and what we have under the ALF condition are indicated in the yellow, where we actually allow flies to feed and have food access during the daytime. And then during the nighttime, they're also given food access and they're able to eat anytime that they want. Ad libitum feeding just basically means feeding at will. And this is done in a 24-hour cycle. And the other condition that I'll be introducing is the time-restricted feeding condition. And this is basically given a 12-hour feeding window for the fruit flies. The fruit flies are what we work with, and they're basically only given food during the first 12 hours of the daytime, but during the nighttime, they're actually switched into a vial with just agar just to prevent desiccation. So one of the main overarching hypotheses of my project and my goal is basically that muscle dysfunction can be caused by metabolic challenges such as aging, circadian disruption, and obesity. And this can potentially be attenuated through homeostatic feeding fasting rhythms imposed by interventions such as time-restricted feeding. And by imposing time-restricted feeding, this can modulate key gene expressions that are potentially involved in metabolism, which can then feed into actually regulating muscle function. So I don't say this without any preliminary data. So I'd like to show a little bit of what we've accomplished in the past. We've actually imposed these fruit flies under different metabolic conditions, and we've imposed this time-restricted feeding. Now, here are 4 different metabolic parameters that we measured: weight, triglyceride, glucose, and an insulin resistance marker. And from what we can see, going from left to right, the dark blue is basically a wild type control under a regular diet. This is just our control. And then we also place this control onto time-restricted feeding. We did this into 2 additional obese conditions, which is a high-fat diet-induced condition. And then we also have a genetic mutant, which is SK2, part of the genetic-induced obesity. So with these 2 different obese conditions, you may not see so much improvement in the healthy wild type control, but once you actually impose these metabolic challenges, you can see that there's a significant improvement, at least in body weight from high-fat diet and genetic-induced obesity. And you can see that in triglyceride, glucose, and also in the insulin-resistant markers. Interestingly, we further bolstered this by also measuring the physical performance of these fruit flies. We measure 2 different assays here. We do a flight index and we measure a geotaxis assay. And interestingly, the time restricted feeding, again, once again, is able to show this improvement, or at least this attenuation of phenotype within these different metabolically obese groups. You can see that in high fat diet, ALF versus TRF, there's this drastic improvement in flight index. The higher the flight index, that just means the more capable they are of actually flying up. They have a better trajectory. And we were able to show this within different age groups, basically, that even as you age, you're able to retain this flight performance. Even in the control with the later age groups, you're able to retain this performance. And we're able to show this in both male and female conditions as well. So that's the flight index, and this is the actual climbing ability. And we measured here the different percentages that are able to reach a certain threshold. And the way geotaxis is, basically, if you perturb a vial of flies, they will always have a natural tendency to go against the vector of gravity, so they will climb as high as they can. And given 10 seconds, how many of them are able to climb a certain threshold? And that's what this above percent threshold means. So within our TRF conditions, again, we see a similar pattern where a lot of the TRF conditions are able to perform and outperform the ALF counterparts. So the next thing that we're currently interested in, so we've seen a lot of these different attenuations of metabolic challenge, but why is it doing that? That's the next question. And the way that we're addressing this is what we have here is a DIO, diet-induced obesity fruit fly, and we have a genetic-induced obesity fruit fly. We go ahead and we split them off into both ALF and TRF 7 days a week. And as shown, the ALF conditions showed metabolic disease in skeletal muscle while they showed improvement in muscle performance. And furthermore, what we'd like to do now is we'd like to isolate in a temporal manner muscle transcriptomics by collecting different fly muscle at different time points under these different ALF TRF conditions in obesity model. And furthermore, whatever genes we find from that, we then like to do a genetic validation by ordering genetic knockdowns of these specific genes that are changed in TRF. And we'd like to see whether or not these genes are actually important for muscle function. So what we found here is that in common both DIO and GIO, we found that 3 specific genes were upregulated, and that's GNMT, SARDH, TDH. These 3 genes can have a major role in 1-carbon metabolism, and they can also have other downstream effects that might regulate different pathways that I'll go over in a little bit. Furthermore, DGAT2 is a key triglyceride synthesis enzyme regulator, and that would make sense. So basically, if you're lowering DGAT2, that means you're actually regulating and reducing the amount of triglyceride that's being produced. So we then moved on away from common upregulated genes to more exclusive model-specific genes. And what we found was that under diet-induced exclusively, we found that purine biosynthesis was majorly upregulated, while in GIO, genetic-induced obesity, we found that AMPK signaling was more specifically upregulated in addition to downstream pathways such as glycolysis, glycogen metabolism, and ETC. So interestingly, GNMT can, through other previous literature, can actually play a role in both of these purine biosynthesis and also AMPK signaling, in that GNMT has been shown by other literature that it can actually help activate purine biosynthesis. And biosynthesis can actually regulate ATP energy, and it can maintain energy balance so that the muscle can have enough ATP for energy contraction. Additionally, AMPK, of course, AMPK, there's many downstream targets. There's even TCA, and TCA can have beta fatty acid oxidation. There's also glycolysis. A lot of this are also involved in energy and ATP management. So I think that it's very interesting that ATP energy balance might actually be quite instrumental for TRF's ability to mediate a lot of the improvements that are being seen in muscle function. Moving forward, we took some of these genes, GNMT, SARD. Actually, we took all of these genes, but for the sake of time, I'm only showing you the common genes here. But we've taken GNMT, and what we can see is that upon knocking down these genes using a UAS-Gal4 mechanic, we are able to see that there is this reduced flight performance, even in just regular wild type flies. So it's indicating that GNMT can have a very important role in muscle function as well. We did this also for the other common genes, and again, they showed a very similar trend. Furthermore, what we did was we took these common genes and we then performed a knockdown, and we wanted to see whether or not we can still instill TRF mediated benefits, and it seemed to abolish a lot of the TRF benefits that we previously saw. This potentially indicates that these genes are very important for TRF's ability to really enhance and attenuate these metabolic obese challenges and improve flight index. Finally, we also showed the down-regulated gene of DGAT2 and that triglyceride synthesis can have an effect. So this would be the opposite. Now you would expect improvement, because if you knock down DGAT2, similarly as what's seen in TRF, you're able to retain the actual flight index, whereas in control, within the first 3 weeks, you see that there's not much significance. Control is relatively healthy within the first 3 weeks, but after 5 weeks, age begins to decline, and you can see the sarcopenic effect, where you can now see this deterioration in flight index, but a lot of this can still be preserved with DGAT2 knockdown. So with that, I'd like to give a little bit of summary of what I've seen so far. So TRF is very interesting. It showed improved muscle flight performance, in addition to also improved metabolic profiles in attenuation of obesity. Furthermore, we collected transcriptomic data within fly muscle, and we've shown that TRF can have commonly upregulated genes that are potentially involved in 1-carbon metabolism, while downregulation was involved in triglyceride synthesis. Exclusively in DIO, we found that purine cycle seemed to play a major effect. And again, purine cycle, although it's involved in things such as immune-related defense, bacterial response, it also has a role in ATP and energy management. And we think that this energy ATP management might be quite instrumental to muscle function. Additionally, in GIO, AMPK signaling can also be affected. And it's very interesting that both of these seem to have a common denominator, in which that ATP and energy balance is all very instrumental for TRF improvement. Finally, we tested this hypothesis by doing an RNA knockdown, and we wanted to see whether or not we can actually still improve these flies with a knockdown in these genes. And furthermore, we saw that TRF benefits were abolished. So this is an ongoing project, and my future directions is I'd like to explore the other metabolic challenges that I previously described, such as circadian disruption. In addition to that, I'm going to be pursuing an aging model that is inclusive of more time points. Furthermore, we're also looking into using biochemical assays such as ATP, triglyceride, glucose to really drive home the point of energy balance being critical for this TRF-mediated improvement. Furthermore, we'd like to see if there's any muscle integrity changes using cytology. And we would like to see whether or not different metabolites from our proposed pathways are being changed within these different conditions. So with that, I'd like to really give a big thanks to my team. Everyone in our lab is really amazing. I'm really happy to be a part of this lab. And thank you very much for Dr. Girish Melkani's tutelage. I'm constantly under his tutelage, and it's really helped me a lot. And also my collaborators and some funding information. And with that, I'd be more than happy to take any questions from anybody.
Right. That was exactly 15 minutes. So, but fortunately, the other speakers were a little bit early, so we do have time for 1 or 2 questions before the hour's up. I will say I'm always very impressed by how useful the fly model is. I know a lot of people, it's not usually the model we come to, or at least I don't come to first for metabolism, but it's great to see the same pathways coming up. The question I have for the muscle work is, in mammalian systems, the fiber type really makes a difference for metabolism, like a glycolytic gastrocnemius versus an oxidative soleus, something along those lines. Are you able to get to that level of detail in the fly model? And if so, what muscle type equivalent are you looking at in these studies?
It's a very good question. That's a very relevant question. So the current flight muscle that we're currently looking at, it's called the IFM, or it's rather called the indirect flight muscle. And that's basically just the major muscle group found in adult flies. But in addition to that, we're also looking at the climbing muscle. So that's why we test, actually, I didn't explain this, but we test two different assays. One is the climbing muscle and one is the flight index. And that's just because that's what comprises the muscle groups of the Drosophila. And if you actually look at the similarities between human and flies, the muscles are actually really similar, even down to the actual small muscle fiber types. You can see that the myofibrils and sarcomeric structure are actually very, very similar.
Great. Thank you so much. And please, everybody, join me in thanking all four of our speakers today. You, of course, all did great jobs again, and are really show what a great diversity of research our department has. Thank you all so much. And I please hope you will all join us again next Thursday. Thank you.